In this lecture, we will talk about the early development of mammals. In most mammals, holoblastic cleavage is modified to accommodate the formation of a placenta, which enables the embryo inside of the mother's body. The placenta is an organ containing tissues and blood vessels from both the embryo and the mother. Gas exchange, nutrient exchange, and waste elimination all take place through this placenta. The mouse has been, as an embryological specimen, the model organism of choice and is the subject of many studies involving genetic, surgical, and developmental biology manipulations. In addition, the mouse has been the first mammal in which the entire genome has been sequenced, so it's become a very valuable animal for being able to understand specific genes and what those specific genes do through creating knockout mice in which the gene is disabled and then you can study the effects. So the mouse has really become a very valuable animal, and many studies in cellular, molecular, and developmental biology have been done on this species. Now, as you can see, the mouse is an amniote. We've discussed earlier the chicken, which is also an amniote, along with reptiles. The mouse is an amniote. It's also a mammal, of course. There are a couple of branches of mammals. There are the monotremes, the egg-laying mammals, and there are the placental mammals, and we'll concentrate on the placental mammals. If we look at a diagrammatic representation of what a bird looks like, we've already done this in an earlier lecture, you may recall, that it has structures, and particularly the embryo itself is very similar in appearance to the mammal. It has structures which allow it to maintain the moisture inside of, in this case, this shelled egg. Reptiles also have eggs that are more leathery, but you can see there are comparable parts in the mammalian system, some of which are not functional to any great extent. For example, the yolk sac in the bird is a major organ which allows the bird to get nutrition. In mammals, this has really become a vestigial structure, and instead of using the yolk sac for nutrition, the placenta is used for nutrition. In other words, the mother is providing the food and, of course, the oxygen as well into the embryo, something that the yolk sac also does, is to provide oxygen to the embryo through the vascular system that develops. The allantois in the bird is what collects the waste materials from metabolism, particularly noxious nitrogenous wastes that come from metabolism. There is a structure that is the allantois in the mammal as well. However, again, this does not have a major function at all, if any. And again, the placenta is what allows for the dissipation of noxious waste materials that come from the growth and metabolism of that developing embryo. If we talk about early mammalian development, this happens to be a uterine horn from a human. Here we have the ovary, and we have ovulation that can take place. Once the ovum matures, the fimbria of the uterine tube picks up the ova, and in this infundibular area, this very high up area of that fallopian tube, you can see that fertilization takes place. It's inside of a membrane called the zona pellucida. Now, the zona pellucida is pretty important because what that does is to keep that egg isolated and the developing zygote isolated, so that as it passes down through the uterine tube, it does not attach to that uterine tube. If that zona pellucida were not there, it would allow the attachment, and you would then get what is known as a tubal pregnancy, which is a very dangerous sort of thing, because this developing zygote will really attach anywhere that there are potential blood vessels. When it hits the morula stage, then it basically comes out of the zona pellucida and will then embed in the uterus, and development can start to take place. Let's talk now about cleavage. Mammalian eggs are among the smallest in the animal kingdom, making them hard to manipulate experimentally. The human zygote, for instance, is only about 100 microns in diameter, barely visible to the naked eye, less than 1/1000th the volume of a Xenopus laevis egg. Also, mammalian zygotes are not produced in numbers comparable to sea urchins. or frog zygotes. A female mammal usually ovulates fewer than 10 eggs at a given time, so it is difficult to obtain enough material for biochemical or molecular studies. As a final hurdle, the development of mammalian embryos is accomplished inside another organism. It is inside the mother's body, and so it makes it difficult to get to, unlike studying xenopus or zebrafish, in which the entire development of their embryology takes place outside the body. In this slide, we see a uterus as well as an oviduct, and we see the pathway of a fertilized ovum going down, getting ready to be implanted into the uterus. Let's look now in a little more detail at the type of cleavage that takes place in mammals, which is really pretty unique among all of the vertebrates, in fact, among the entire animal kingdom. Cleavages in mammalian eggs are among the slowest in the animal kingdom, taking place some 12 to 24 hours apart. Meanwhile, the cilia of the oviduct is pushing these eggs toward the uterus, and the first cleavage at the two-cell stage is taking place in that uterine tube. Several other features distinguish cleavage, and one of those is that the first cleavage is normally meridional in its division, and the second cleavage is equatorial. Let's compare the cleavage we see in such organisms as echinoderms and amphibians to what we see in mammals. In echinoderms and amphibians, we see this kind of radial cleavage, where the cleavage in early stages ends up with similar kinds of cells being formed. And that's true in the mammal also. However, in the case of the mammal, the first cleavage is usually done in a meridional plane, like this, and then in the second cleavage you will get another meridional division, but then on the other side you will get equatorial. So during the second cleavage, one of the blastomeres will divide meridionally, and the other blastomer will divide equatorially. And so you get what's called rotational cleavage, and that is unique. Another major difference between mammalian cleavage and most other embryos is the marked asynchrony of early cell division. Mammalian blastomeres do not all divide at the same time. Thus, mammalian embryos do not increase exponentially from 2, 4 to 8 cells like we're used to seeing in other metazoans, but instead contain odd numbers of cells. Furthermore, the mammalian genome, unlike the genomes of rapidly developing animals, is activated during early cleavage, and zygotically transcribed proteins necessary for cleavage and development are made very early then. And you can see that that would be the case because in mammals there's very little cytoplasmic material to contain the RNAs and other controlling factors for early development, so the zygotic nuclei must handle this job at the very early stages of cleavage. Although the maternally encoded proteins can persist through most of the cleavage stages and play important roles in early development, in the mouse and goat, the activation of zygotic genes begins in the late zygote and continues through the two-cell stage. In human, the zygotic genes are activated slightly later, around the 8-cell stage. But you can see in both cases, this is very, very early compared to what we have seen previously in amphibians and the other vertebrates. Another phenomenon that is most interesting is compaction. Now, this shows the cleavage of a single mouse embryo in vitro, so this is in vitro development. One of the most crucial events of mammalian cleavage is compaction. What we mean by compaction is that here we have a 2-cell stage mouse embryo, it goes to 4-cell stage, then to 8-cell stage, and between the 8-cell stage and during the 8-cell stage, the cells begin to compact and become very closely associated. And that's really one of the most crucial events of mammalian cleavage, a phenomenon of compaction. Most blastomeres through the 8-cell stage form a loose arrangement, and then as we get to that 8-cell stage, cell adhesion proteins will form, which pull these cells tightly together. In fact, it's the E-cadherin. You recall E-cadherin being for embryonic cadherin. And the blastomeres gradually are pulled tightly together, and as they're pulled tightly together, they form a very compact ball of cells. The cells continue to divide, producing an inner layer of cells and also then an outer layer. And so this becomes the morula stage. The outer cells are called the trophoblast, and the inner cells of this are called the inner cell mass. The outer cells will essentially become extraembryonic membranes and contribute to the yolk sac and allantois, some of those structures that are vestigial actually in the mouse. The inner cell mass will give rise to the embryo body proper. In other words, the actual embryo. And that's happening in here, and as development progresses between here and here, you start to get a cavity form because of secretion of fluids that will allow for that cavity to form. And so here you're at the blastocyst stage right here, and this is actually the stage that will embed in the uterus and establish a placenta and start to get nutrients from the maternal blood circulation. So, just to summarize, you're going to the 8-cell stage. You get a compaction here. This is the morula stage. It will form a cavity inside through the secretion of fluids, and eventually the inner cell mass, which is going to become the embryo body proper, will be pushed to one side, and that will develop as the new embryo, in this case a mouse embryo. This is an electron micrograph, so you can see some of these structures a little better. Here's the cavity. These are the trophoblastic cells around the outside, and here are the cells in the inner cell mass that are totipotent cells. Totipotent means that they can develop into anything, including a whole new organism. Now, you recall when we looked at the slide of the uterus that as the zygote was migrating down through the fallopian tube, it reached a point where it no longer had a zona pellucida. The zona pellucida came off so that it could now have its cells exposed to the uterine wall. And here you see an embryo at this stage hatching from the zona pellucida. It's getting ready to attach to the uterine wall. Here, now, these are mouse embryos. Here you can see these mouse embryos in the blastocyst stage. That's the stage when they actually will attach. You can see 3 such embryos here, and they are ready to form attachments to the uterine wall so that the placenta can form and they can begin to get the nutrients and continue their process of differentiation and forming into a new individual. This is actually in a rhesus monkey. This is an early stage of a blastocyst that has attached now to the uterine wall. Here you can see the trophoblastic layer that will give rise to, among other things, extracellular membranes, the chorion, and so forth. And here's the inner cell mass that will, for the most part, give rise to the embryonic body proper. And so now that's initially attached, and it will eventually embed into the uterine wall and form the placenta, which will then allow the nutrients to be provided from the mother to the fetus, and the waste products from the fetus to the mother, along with an exchange of oxygen and carbon dioxide. Let's now talk briefly about twinning. The early cells of the mammalian embryo can replace each other and compensate for a missing cell. The regulative ability was first demonstrated when one cell in a 2-cell rabbit embryo was destroyed and a totally normal rabbit embryo was produced. The regulative capacity of the early embryo is also seen in humans. Human twins are classified into 2 groups. One is the monozygotic group, in which a single egg is fertilized, and then somewhere along the line, that egg or zygote will split into two genetically identical zygotes, which will end up forming identical twins. Dizygotic twinning is simply a matter of where two eggs are fertilized and they independently grow, certainly simultaneously in the same uterus, but genetically they are not any more closely related than would be siblings. But if you look at monozygotic twinning, you can see that if the embryo of the zygote splits very early, you will get two trophoblasts formed around here, and you will end up with two chorions that have placentas that are separate. And as they go into embedding in the uterus, then you will get two complete chorionic sacs, two separate embryos. They are genetically, of course, identical yet because they started to be from a two-cell embryo that split, but they split very early. If the splitting takes place a little later, let's say that it takes place after they're within that trophoblastic ring, then both will end up on the inside of the chorion, and they will have separate but joined placentae, and they will again be in separate individual amnions, but they will be in a single chorion. And if it happens quite late in development, then they're within the single blastocele, and they're joined early so that they form a single amnion. At that point, then, when they are embedded in the uterus and form the placenta, they have a common placenta. They have umbilical cords that are separate, of course, since they're two individuals, and at this point, they of course are identical individuals again, but within the same amniotic sac. Let's also talk about some experimental studies in which chimeric individuals have been formed. Now, sometimes when, say before we move on to that, sometimes in this particular instance, if the embryos have grown too far, and they may have parts that have formed from the same group of cells, they could become conjoined. So you could get twins or multi-headed individuals. You could get twins that are joined together in various parts of their body, or you could have an individual that has a body with two heads. So there are various things that can happen in these kinds of situations. Let's look at the production of chimeric animals, and of course mice are used as the model since they're the animals that are of experimental choice. There are many genetic versions. If you take and mix cells in the early stages of cleavage, 4 or 8 cells or so, mix them together, dissociate the blastomeres, and make a chimera, let them form into a blastocyst in vitro, and then implant those blastocysts into a foster mother, you will get an organism that appears normal except that it's a chimera. It has body parts that are made from the different embryos that have been fit together, and so here that's expressed in seeing the different colored fur. Here's another example. This is a different set. This is a female mouse who has mothered these 3 offspring. This mouse was a chimera derived from 3 different animals, 4 or 8 cell stages of 3 different animals in this same manner. It was a black mouse, a white mouse, and a brown-colored mouse. This female was then mated to a recessive white male mouse, and what she produced were totally black mice, totally brown mice, and of course the recessive white mouse here. And what that showed was that her germ cells had been derived from all 3 of those individuals and functioned completely normally in her body. So this is an example of chimeras, and this is what can be done from this kind of experimental developmental biology manipulation. Let's now just very quickly review the tissue and germ layer formation in an early embryonic human. This shows an embryo at day 5, the blastocyst stage, and now it's gotten outside of its zona pellucida, so it's ready to implant into the uterus, and here you can see that it has formed a hypoblast and epiblast, and The extensions are coming in to the beginning of the formation of what will become the placenta. And as development progresses, there's an erosion of the uterine wall, which readily accepts the embryo coming in, starts to cavitate, and so it starts to form an association, close association, with the developing fetus, and it forms extraembryonic membranes that are tightly associated between the mother and the developing embryo. And if we look at these embryos, what's very interesting is that in spite of the fact that these embryos have virtually no yolk, they're still flat. They're flat as if they were sitting on top of a big yolk, like in an egg or in a chicken egg or in a reptilian egg. And so this is where evolution hasn't perhaps been completed yet, because these embryos really don't have much yolk, and yet they're shaped as if they were sitting on a large amount of yolk and not able to divide completely all the way through. In any event, this is a remnant of being derived from reptiles and avian species as offshoots of similar kinds of ancestors. Here we see the primitive streak, here's the ectoderm forming, here's the embryonic mesoderm, and the embryonic endoderm. It's virtually the same as in the chicken or in the reptile. If we look at this flow sheet, we can see that the blastocyst gives rise to the trophoblast on one side, cytotrophoblast, and syncytiotrophoblast. These are all going to be extraembryonic structures, but the blastocyst gives rise to them. If we take the other side, then the blastocyst also gives rise to the inner cell mass, which is in here, this inner cell mass that's going to form the embryo proper, the inner cell mass. And the inner cell mass will give rise to primitive endoderm, the hypoblast, extraembryonic endoderm, which will also contribute to some of the extraembryonic membranes, in this case the yolk sac. The epiblast will then give rise to the amniotic ectoderm and the embryonic epiblast, and these then will form the primitive streak and form the three germ layers, the embryo proper, the ectoderm, the endoderm, and the mesoderm. And this mesoderm then will also line the inner part of some of the extraembryonic material to form extraembryonic mesoderm. Let's now look briefly in a little more detail at this inner cell mass, which is going to form important components, including the embryo itself. Here's a mouse embryo at day 3.5, early blastocyst stage, showing the random expression of a couple of genes, NANOG, and that's the blue that indicates the presence of an epiblast, and GATA6, that's for visceral endoderm. It's in the inner cell mass here. At this stage, 3.5 days, it's pretty much a random mixture of these cell types indicated here. The first segregation of these cells then within the mass forms two layers. And the lower layer is in contact with the blastocele and is called the primitive endoderm, which we don't see here yet, and it's homologous to the hypoblast of the chick embryo. So let's look at that. We have the hypoblast here now. It's 24 hours later. This is at 4.5 days. Here's the inner cell mass here then, and we have the hypoblast here, and so there's now been a segregation between the trophoblastic material and inner cell mass and the hypoblast. Just in that short time, this kind of segregation has taken place. And if we look at the amnion structure and cell movements during gastrulation, it's clear that the primitive endoderm, like the chick hypoblast, is an extraembryonic layer and does not provide cells to the actual embryo. If we look at the amnion structure and cell movements during gastrulation, here's extraembryonic material here. This is going to form the placenta. Here's the embryo proper with a bilaminar germ disc. Here's the amniotic cavity, and here's the yolk sac. And you can see that these cells are forming. This is going to be the embryo proper. Within the amniotic cavity, and look down on the top of this, and we could see the cells migrating. We would see that we have a node here comparable to Henson's node, comparable to the dorsal lip of the blastopore, where involution is taking place. You see the cells are going in and going between the hypoblast and the epiblast. When that happens, the embryo is starting to form. We're getting the embryo formed. We're getting the various mesodermal layers. The primitive groove is here. The endoderm is forming here. The mesoderm would be above that, and here you can see the mesoderm now here. Endoderm forms a layer or primitive gut, and here's the node. So this is gastrulation then taking place in the mammal. Very similar to what is happening in the avian species, and for that matter, what is happening in the amphibian and fish species. The same relationships, little different format in terms of structure, but not really that different because the node is comparable to the dorsal lip of the blastopore or to the embryonic shield, for that matter, in the fish. And that's what allows the 3 germ layers to be formed. That's what allows embryo to be formed, and that's really what allows this individual to become a new organism. I might mention that once this amniotic cavity becomes formed, it fills with fluid to provide that fluid environment that is required for the amniotes. Let's just look a little bit more detail at the placenta. You recall that the chorion formed while the embryonic epiblast is undergoing cell movements reminiscent really of those seen in reptilian and avian gastrulation. The extra embryonic cells are forming the placenta, which is very distinct for mammals because no other species have placentas. And this mammalian set of tissues, this is what really enables the embryo to survive within the maternal uterus. It's to allow that exchange of oxygen and carbon dioxide and food sources and to get rid of noxious materials from metabolism and digestion of some of the food. The extra embryonic mesoderm joins the trophoblastic extensions and gives rise to the blood vessels that carry nutrients from the mother to the embryo. The narrow connecting stalk of extra embryonic membrane and mesoderm that makes up that membrane links the embryo to the trophoblast and eventually forms the vessels of the umbilical cord. The fully developed extra embryonic organ, consisting of trophoblast tissue and blood vessel containing mesoderm, is the chorion. And it fuses with the uterine wall decidua, and this then forms the placenta after fusing with the uterine wall. Here you can see the amnion. Here's the chorionic cavity here. This happens to be the yolk sac right here behind the developing human embryo in this case. I should mention that beyond actually providing nutrients and oxygen, there's also an immunological aspect to the placenta. There are certain kinds of immunological organs, suborgans in the placenta that produce hormones such as progesterone and other things that help the uterus maintain pregnancy, accelerate mammary gland development in the mother, and also to keep certain kinds of disease processes from entering into that area of the uterus. If we look at this diagrammatically, this kind of summarizes what we were saying before here, the chorionic villi that are coming down into the uterine wall. Notice that there are 2 umbilical arteries, and notice that they are blue. They're umbilical arteries, so they're coming out of the fetus, spent oxygen, and they're coming back in to be rejuvenated, and the rejuvenated blood will come back in through the veins. So the oxygenated blood comes back in through the veins, and the spent blood comes back to the uterus for replenishing coming out of the fetus. I'd like to now talk just very briefly about mammalian axis formation. It's very similar to what we see in the other organisms, and we've already talked about it in some detail. But let me just remind you that, of course, in this the highest vertebrate, there is patterning, just like there is in the lower vertebrates. Here we see that it's a gradient situation in terms of patterning, and we can see that here's the embryo area here, the ectoderm, mesoderm, and endoderm, and there are various growth factors and cascades of transcription factors that will affect being an anterior component of the organism versus posterior. And so if you have the FGFs and WNTs and BMPs, you would get posterior area, and if you have other antagonists to these, then you get anterior. The BMP WNT antagonists to neutralize the effect of these. You will get the anterior components of the embryo. This is simply a diagrammatic representation which shows staining intensity for the various, this happens to be FGF8 in terms of patterning in the embryo. And if we look at some of the other components that are a part of this whole cascade, all of them really lead down into directing the Hox genes into anterior and posterior patterning. And if you recall, the Hox genes have been notorious in all of the animals that we've talked about, even back to the Drosophila, when we talked about patterning and when we talked about anterior versus posterior. So in fact, let's look at that. It's quite interesting. If we look at, as I mentioned, the anterior-posterior patterning, Hox is key code during development to make part of the embryo anterior, part of the embryo posterior, and have a transition in between. And like I said, all of the known mammalian genomes contain 4 copies. When it comes to anterior-posterior patterning, the Hox code hypothesis is really an important component when you talk about anterior-posterior patterning. In all vertebrates, the anterior-posterior polarity becomes specified by the expression of Hox genes. Same is true in Drosophila. All of the known mammalian genomes contain 4 copies of the Hox complex per haploid step, and these are the Hox genes indicated here in the mouse, and you have various derivations of the different Hox components. And if you look at Drosophila, you find similar kinds of components in the patterning Drosophila compared to the patterning in the mouse. Similar kinds of genes. The ABDB, a Hox version gene, also in the posterior end, and genes that go forward similar to what we're seeing, components to what we're seeing in the mouse. We also see a similar kind of representation of patterning with Hox in the chick and mouse, and of course we would expect to see that. And as we look at the structures and compare them, it's quite interesting that there are certain Hox genes that are associated with certain structures. For example, if you compare the cervical area of the chick with the cervical area of the mouse, Hox5 is very important in that. And Hox5 has a representation on a number of these vertebral areas, making the cervical area longer, chick, compared to the mouse. And you compare them, the thorax. Again, the same gene is regulating that length, an anterior-posterior length, lumbar area, sacral, and so forth. So the Hox genes are critically important when we talk about vertebral patterning along the anterior-posterior axis in vertebrates. If we talk about left-right asymmetry in the developing human, we've already talked about that. Things start bilaterally symmetrical, but really we become asymmetrical. If we look, this is a cross-section of a human embryo at 5 weeks, 7 weeks, and 11 weeks, and you can see that the liver is moving to the right and other structures are moving to the left. The spleen and pancreas, the heart moves to the left. There are other forms of asymmetry in terms of the vessels coming out of the heart. The aortic arches, superior and inferior vena cava, the branches off of the aortic arch. If we look at the lungs, the right lung has 3 lobes, the left lung has 2 lobes. The left lung doesn't occupy as much volume as the right lung, but what you have instead is the heart forming in this region. And so development has accommodated an increase in heart tissue and a decrease in lung tissue to make things fit in this asymmetric. And with that, we've covered what I wanted to have you hear about in developing mammals. I think what you realize now is that when we talk about embryonic development and we talk about the vertebrates, although on the surface it seems that there are significant differences, really when you get down to the mechanisms and look at things like the dorsal lip of the blastopore and the primitive streak and the nodal tissue, that sort of thing, you realize that all of these things are really the same, and they function in the same way, they have the same genes operating, and they cause the 3 germ layers to form in virtually the same way. Little different structure. One embryo is flat initially and one is more rounded, but in fact these organisms are very, very similar. And so that's something to keep in mind as you're learning about developmental biology and embryology, that there are many, many similarities along with, of course, some of the differences that we see.